Hi there, and welcome to the run through of the 2025 National 5 Biology past paper. We're going to start off with section 1, the multiple choice section, which is worth 25 marks. So there are 25 questions in total. Now, in this section of the paper, you will be choosing from 4 options, A, B, C, or D, and you'll be marking it onto a grid with a pencil. We'll run through it on the computer here, and we'll talk through not just the answers, but also the reasons for the answers and the working for some of the questions as well. Question 1, which of the following structures would not be, you'll notice that in bold, found in a typical plant cell? So we've got the choice of chloroplast, cell membrane, plasmid, and mitochondrion. Now, hopefully you've learned that the chloroplast is part of a typical plant cell, so we can eliminate that one. The cell membrane is found in all cells, so therefore we can eliminate that one. The mitochondrion is found in most cells, including the plant cell, so we can eliminate that one. And C, the plasmid, is found only in the bacterial cell. So we know from our study of the bacterial cell that the plasmid is only found there and not in the typical plant cell. So the answer to the first question there is going to be C, plasmid. Question 2, potato cylinders were placed into different concentrations of sucrose solution and any change in mass was recorded. The table shows the initial sucrose solutions inside the potato cells and the concentrations of the solutions they were placed in. Which potato cylinder will gain the most mass? Now, aside from being one of the most exciting experiments you can do is, probably remember it from class where you bore up some potatoes and pop it in some water and leave it for a few days. Aside from that aspect, what this experiment does show is it shows osmosis taking place within plant cells. And so essentially what they're asking you here in this question is, when it says which potato cylinder will gain the most mass, you're basically looking for the potato cylinder which gains water. Okay, now we know that osmosis is the movement of water molecules from a higher water concentration to a lower water concentration. So what we have to do here is we need to work out where the higher water concentrations would be and where the lower water concentrations would be. That's the first step. Now, if we look at the two columns, what it gives us is the sucrose concentration, not the water concentration. So what you have to do is you have to do a quick conversion back to water concentrations. So if we do a quick run through of all 4 options that we've got, A says that the sucrose solution inside the cell is 0.31 and the sucrose solution outside the cell is 0.45. So that tells us that we're going to have 99.69 inside the cell and 99.55 outside the cell in terms of water concentration. So 69 being bigger than 55 means that the water is going to move from inside the cell for A to outside of the cell, which means the cell is actually going to lose water. Therefore, it will not gain the most mass. Option B, we can see that the sucrose solutions are the same both inside and outside the cell. So therefore, there'll be no net movement of water molecules. For C, we can see that we get 0.31 again inside the cell and 0.25 outside the cell. Now this time we've got 99.69 inside the cell and 99.75 outside the cell. So we actually have a higher concentration outside of the cell this time, and that means that water is going to move from the higher outside to the lower concentration inside. So it is actually going to gain mass for C, but the question asks us which potato cylinder will gain the most mass. So if we look at D, we're going to have 0.35 inside, which is 99.65 water, and we're going to have 0.21 outside of the cell, and that's going to be 99.79. So 79 being bigger than 65 means that the water concentration is higher outside, and therefore water is going to move into the cell for D. And because the concentration gradient is greater for option D than it is for option C, more water will move into the potato and therefore it will gain the most mass. Okay, so we're looking for the greatest difference in concentration gradient there, and D has a slightly greater difference than C does. So that's why D would be the answer there. But that is a tricky question, and it involves a wee bit of working out there. Question 3. In a section of DNA, 34% of the bases are thymine. Which row in the table shows the percentages of the other bases in this section of DNA? So we know that, or hopefully we know, that thymine is complementary to adenine, and therefore we would expect the percentage of bases in this section of DNA to be the same for adenine as it is for thymine. And if we look at the table, options B and C both show that. So we know the answer has to be B or C. Now, if we combine the thymine and adenine together, that's 34 times 2. So that's going to be 68 for adenine and thymine in total. So therefore, that leaves us with 32 that are going to be shared between cytosine and guanine, because those two are complementary to each other. And with 32 divided by 2 is going to be 16. So there's going to be 16 cytosine and 16 guanine. So the answer we're going to look for here is C on this one. 34 adenine, complementary to thymine, which we're told is 34. That leaves over 32 to be shared between cytosine and guanine. So that's 16 each. Okay, so the answer for number 3 is going to be C. Question 4. Lipase is an enzyme that speeds up the breakdown of fats, producing an acid that decreases the pH. 3 test tubes were set up to investigate the action of lipase on fat and milk. So we get test tube R, which is milk and lipase, test tube S, which is milk and water, and test tube T, which is milk and boiled lipase. Note the boiled. The pH of each test tube was recorded at the start of the experiment, and then again 20 minutes later. In which of the following test tube or tubes would the pH stay the same? So if the reaction is taking place, if lipase, the enzyme, is breaking down the fats, that means that the pH will become more acidic. That will decrease the pH. Okay, so if we look at our milk and lipase, now I would imagine that everything's going to function properly here, that the lipase is going to break down the fat in the milk, and therefore it will become more acidic. Okay, so therefore the pH should decrease in R. In S, we've got milk and water. So we're lacking the enzyme lipase this time. So I would imagine the pH would stay the same in this one. And then lastly, the milk and the boiled lipase. So we have the enzyme present this time, but because it's been boiled and therefore denatured, it will not be able to catalyze or speed up the breakdown of the fats, and therefore it will not become acidic, and therefore the pH should stay the same. So we're looking at S and T, which is option D for this particular question. Okay, R would become acidic because the lipase would break down the milk, but S and T would both stay the same. Question 5. A 100-gram sample of hemoglobin was found to contain 15 grams of leucine. What mass of leucine would be found in a 5-gram sample of hemoglobin? So a little bit of maths in this one. First thing we have to do is we have to find out how many times 5 grams will go into 100 grams. Okay, we need to scale this down. So if 100 grams of hemoglobin has 15 grams of leucine, how much would there be in 5 grams of hemoglobin? So 100 divided by 5 gives us 20. Okay, so that means that it should be 20 times less for the 5-gram sample of hemoglobin, okay, because it goes into 120 times. So what we then have to do is get the 15 grams of leucine, and we need to divide that by 20. And when you do 15 divided by 20, it should give you the answer of 0.75. Okay, so a little bit of scaling involved, a little bit of maths involved here, but that's how we would do it. 100 divided by 5 gives you 20. And then because we know it's 20 times smaller the sample, then we have to divide the 15 grams we found in the original sample by 20 to give us 0.75. Question 6. A culture of 350 bacterial cells was genetically engineered to produce a human protein. Only 210 cells successfully produced the protein. The percentage success was... So it's telling us there's a total of 350 bacterial cells, but only 210 were successful. So we have to work out 210 as a percentage of 350. So we do 210 divided by 350 times by 100, and that should give us an answer of 60%, which would be B there. Okay, so that's a simple percentage calculation. Question 7. Which of the following builds up in animal cells as a result of fermentation? So again, this is a straightforward knowledge question. There's nothing to work out here. We just either know this or we don't. Is it lactate, ethanol, glucose, or carbon dioxide? Now, we know that lactate is produced in animal cells as a result of fermentation. So immediately we suspect we know the answer there. But we can just eliminate the other ones. Ethanol is produced by plants and yeast during fermentation. Glucose is obviously the initial substrate, the initial thing that's broken down in the first stage of respiration. And carbon dioxide is a waste product in both aerobic respiration and fermentation in plants and yeast. So therefore, is not produced by the animal cells. So the thing that you had to know here was how animal cell fermentation is different from plant cell and fungal cell fermentation. Question 8. The apparatus shown was set up to investigate the effect of temperature on fermentation in yeast. The investigation was carried out at 15 degrees and then repeated at 20 and 30 degrees. The number of gas bubbles produced was counted. Which two variables should be kept constant during the investigation? So the choices are temperature and time left for, the type of yeast and the time left for, the temperature and the carbon dioxide concentration, and the type of yeast and the carbon dioxide concentration. Now, it tells us in the description that the investigation was carried out at 3 different temperatures. So therefore, it doesn't make sense that temperature should be kept constant during the investigation. And because of that, we can rule out A and C. The type of yeast, that makes sense. We would want to keep the same type of yeast because different yeast will undergo fermentation at different rates. And the time left for, that makes sense because if we're counting the number of gas bubbles produced, if we have different times, then that's not going to be fair to compare those two. It will not be valid. And the type of yeast and the carbon dioxide concentration, well, the carbon dioxide concentration is not going to have any impact on the rate of fermentation. So that one is irrelevant as well. So the answer is going to be B for this one. Okay, the type of yeast and the time left for. Question 9. During mitosis, which of the following occurs immediately before the cytoplasm divides? So for mitosis, you have to learn your steps. It's a memorization topic. You just have to learn the steps and know those off by heart for the exam. And if you have done that, then you'll know that it's the nuclear membranes form. Okay, the chromosomes line up at the equator happens earlier. The pairs of chromatids are separated happens just after that. And the chromosomes shorten and thicken happens right back at the start. So the nuclear membranes form is the one that immediately precedes the cytoplasm dividing. Okay, so that's going to be A for question 9 there. Question 10. The function of an interneuron is to carry electrical impulses from. So the interneuron, if you remember the acronym SIM, sensory intermotor. If you remember your SIM card in your phone, that will help you remember that one. Interneuron's in the middle. Okay, so therefore it's going to be connecting the sensory to the motor neuron. And if we look at the options, it's C is the only one that makes sense there. SIM, sensory, intermotor. So the interneuron will connect the sensory to the motor neuron. Okay, it can't be A because you'd be going the wrong way there, going from the motor back to the sensory. And B, the effector is at the very end, beyond the motor neuron. So that doesn't make sense. And for D, the motor neuron to the effector, that happens after the interneuron. Okay, so that one wouldn't make sense either. So it's only C that makes any sense for this particular one. Okay, number 11. It says, which row in the table identifies features of the hormone glucagon? So the old rhyme that I teach my classes is, when the glucose has gone, release glucagon, which basically means the glucose has gone, i.e. the glucose levels have fallen. Okay, so glucagon is released by, the site of production is the pancreas. The target organ, the place where it has an effect, is the liver. Okay, so that means we're looking at either A or C based on that. Then the effect of glucagon is because the glucose has gone in the blood, we want to make glucose, we want to release glucose back into the blood. And the one that shows that is A. Okay, glycogen getting broken down into glucose, which is then released back into the blood. All right, so that's going to be A for number 11. Number 12, the table shows the number of people recorded as having diabetes in Scotland over a 5-year period. Okay, you've got your 5 years there, and you've got the number of people with diabetes in the table. Now, you have to predict the number of people likely to have diabetes in year 10 if the average yearly increase continues. Okay, so for this question, we have to calculate the average yearly increase. Okay, and then we have to continue to add that. Now, between years 4 and 5, we have an increase of 6,000 there, from 298 to 304,000. Okay, so there's an increase of 6,000. Now, we're adding 5 more years to get to year 10. So the 5 years times the 6,000 would give us another 30,000 or so. So therefore, if we're adding 30,000 onto 304,000, that gives us 1,000. And if we look at the the options we have there from A, B, C, and D, 334,000 is closest to B. Question 13. The table gives data on the number of eggs produced and the percentage of eggs surviving to adulthood in 3 different organisms. So we've got the brown trout, the salmon, and the common frog, and you've got their various figures there for the eggs produced, surviving to adulthood, and the percentage of eggs surviving. So the number of eggs that were produced by the common frog was, so you're doing a reverse percentage calculation here in order to find this out. If the number surviving were 23, and that's 5% survived, you have to work out what 100% was. So the easiest way to do this is to do 23 divided by 5, that tells you 1%, and then times that by 100, and that tells you what 100% is. And on this occasion, 23 divided by 5 would give you 4.6, and then you times that by 100 and it gives you 460. Okay, so that's going to be D is the answer for this one. Okay, so 23 divided by 5 times 100, and that will give you your answer there. So it's a reverse percentage calculation. Find out what 1% is, and then if you can find out 100%, which is what the original number would be. Okay, question 14. The graph shows the growth of a fetus. Okay, we can see the length of the fetus changing there as the age of the fetus increases in months there. So it asks us, the average monthly increase in length of the fetus from month 5 to month 9 is. So we look at month 5, and we track up on the graph. The length of the fetus at month 5 is 250 millimeters. The length of the fetus at month 9 is 410 millimeters. So that's a difference of 160 millimeters, but that's across 4 months, and it wants the average monthly increase. So we have to do 160 divided by those 4 months, and that will give us 40 per month. Okay, so the average monthly increase in the length of the fetus from month 5 to month 9 is 40 millimeters. That's going to be B. Okay, question 15. In guinea pigs, short coat is dominant to long coat. A group of heterozygous guinea pigs were crossed, and a total of 72 offspring were produced. Identify how many of the offspring would be expected to have short coats. So for this one, we have 72 offspring in total. Now, if they're heterozygous guinea pigs and they're being crossed, then what we're going to see is we're going to see a ratio of 3 of the dominant to 1 of the recessive. Okay, and it tells us here that short coat is the dominant one. So we'd have a 3 to 1 ratio of short to long here. Now, an easier way to work this out might be to say, well, 3 to 1 is the equivalent of 3 out of 4, or 3/4. And then we can work out and calculate 3/4 of 72. Okay, we do that by dividing 72 by 4 and timesing it by 3. Now, when you divide 72 by 4 and times it by 3, that should give you 54. And that means that the answer would be C in this one. Okay, question 16. The following sequence shows part of the blood flow through the body. Okay, familiar question. We've seen this a few times before. Which row in the table identifies E, F, and G? Now, we know that the atrium comes before the ventricle, A before V, because A comes before V in the alphabet. So we know that E is going to be the ventricle associated with the right atrium, which will be the right ventricle. So that means that our answers are now limited to either A or B. Now, F is the blood vessel that takes the blood from the right ventricle to the lungs. Now, the hint for this is whenever you see lungs, think pulmonary. Okay, the vessels going into and out of the lungs are pulmonary. Now, F is a vessel that's going away from the heart, artery away, AA, towards the lungs. So that's going to be the pulmonary artery. Then G is going from the lungs back to the heart, back to the left atrium. So that's going to be a blood vessel that's going into the heart. So vein in. So that's going to be the pulmonary vein. So once we've worked all that out, we can see the correct answer for this one is going to be 16A. It's going to be A, isn't it? The right ventricle, the pulmonary artery, and the pulmonary vein for E, F, and G. Okay, question 17. 3 students carried out an investigation into the effect of exercise on heart rate. Each student measured the heart rate after completing the same exercises. After each student's heart rate returned to its resting state, they repeated the investigation. Which of the following would improve the reliability of the results? Okay, so let's look at the options and see which one is going to be the best suited here. Increasing the rest period before repeating the exercise, completing the exercise in a different location, changing the type of exercise each time, increasing the number of students exercising. Now, I always teach my students to remember RE. So reliability, repeat. Reliability, repeat. So in this case, if you're repeating the experiment, it's not like a lab experiment where you can just do the same experiment multiple more times. With humans, you need to increase the number of humans that are taking part in the experiment. So from those 4 options, which one makes the most sense there? It's going to be D, increasing the number of students exercising. And if we look at the other options and we rule them out, if we increase the rest period before repeating the exercise, that's going to be changing a variable. Completing the exercise in a different location, again changing the variable, the location. Changing the type of exercise each time, again changing a variable, the type of exercise. The only thing that would increase the reliability of the results is increasing the number of students exercising. So D is going to be our answer here for question 17. Question 18. We have got samples of water that were taken from a river at a sewage outlet and a number of points downstream. The graph shows the oxygen content of the water at different distances downstream from the outlet. Okay, that's fine. We can see that there. So what is the percentage increase in the oxygen content from 250 metres to 1,000 metres from the outlet? So we'll find 250 metres here, and we find that the oxygen content there is going to be 2. Okay? If one big box here is worth 0.1, then one smaller box is going to be worth 0.02. All right, so this will be 250 will be 0.04, won't it? 0.04. And at 1,000 metres, we scroll up here, we see it's all the way up at 1. Okay, so we're going from 0.04 all the way up to 1. So what is the percentage increase? Well, it's the change from 0.04 to 1 divided by the original, which is 0.04. And if we do that calculation and plug the numbers into our calculator, we get 2,400. Okay, and that might seem like a big percentage change, but if you think about the numbers involved, 0.04 and 1, there is quite a big difference there. So that suggests that that sounds about right. Remember, if there's only a 90% increase, that means that it's less than doubled. And obviously 0.04 is more than doubled in this occasion. So that's what gives you a clue, is it will be one of the big numbers. And by doing the calculation, you'll find out which one there. So that's going to be 18C for this one. Number 19, seals and dolphins compete for food. Which row in the table describes the interaction between these organisms? Okay, seals and dolphins are different species, so that's going to be interspecific rather than intraspecific. The way to remember that is interspecific, different species. So there's an ER in different and there's an ER in interspecific. So therefore, we know it's interspecific rather than intraspecific, which is for the same. Same's got an A in it. Intra, it's got an A in it. So we know it's not that. It's interspecific, so that's C or D. And then which one describes the interaction between these organisms? Is it a biotic one or an abiotic one? Well, we know that biotic means living organisms, living things, and abiotic means non-living factors. Seals and dolphins are both competing for food, which is a biotic factor, and therefore it's going to be a biotic interaction. So 19 is going to be C, interspecific and biotic. 20, the diagram shows an experiment used to investigate the conditions needed for photosynthesis. All 4 tubes contain pondweed and water. Okay, we've seen similar experiments to this before in past papers. We've got 4 tubes, 2 in light, 2 in dark, and we've got different carbon dioxide concentrations across them as well. So which 2 tubes should be compared to show the effect of light on photosynthesis? So we think about the purpose of this investigation is to show the effect of light. So that's the thing that should change. But it's the only thing that should change. It's the only variable we are allowed to change. So which 2 of these 4 tubes could we compare where the only difference is between light and dark? So let's have a look at them. So 1 and 3, we have the same carbon dioxide concentration, and the only difference we can see there is between light and dark. And if we look at number 2, we can see the same thing. The same carbon dioxide concentration, but the difference between them is between the light and the dark. So we'll look at the options. Is 1 and 3 an option? Nope, it's not one of the options there. Even though we could compare them, it's not an answer. But 2 and 4 is an option. That's option D for this question. So therefore you'd go for D. Okay, 1 and 2, which is the first option, can't be compared because we are keeping the light the same and the carbon dioxide concentration is different. So therefore that would not be valid. 1 and 4, we've got light and dark, which is good, but we've got 0.03 carbon dioxide concentration with 0.3, so a different carbon dioxide concentration. And then 2 and 3 won't work for the same reason. The carbon dioxide concentration has changed as well as the condition of light or dark. Okay, only one variable at a time can be changed to keep it valid. Okay, question 21. The graph shows the effect of limiting factors on the rate of photosynthesis. So X here, we've got more carbon dioxide, we've got 1.0% compared to 0.1, and the temperature has been kept the same between X and Y. So which row in the table identifies the limiting factors at points X and Y on the graph? So I always teach my students that when the point is on the upward part of the curve or the graph, like X is here, then the limiting factor is what is written on the axis here. So that's going to be light intensity. Okay, if the graph is still going up, the limiting factor is whatever is in the bottom axis here, which will be light intensity. For Y, this point we've leveled off, so it's no longer light intensity that's a limiting factor. It must be something else. And if we compare the two lines, you can see the temperature is the same, so that hasn't changed. But the reason why this line at the top is much higher in terms of its rate of photosynthesis is because of the increased carbon dioxide concentration. So therefore, on Y, the limiting factor is carbon dioxide concentration. So the combination which works in the answer table there is B, light intensity, and CO2 concentration. So 21 will be B. 22, we've got some pyramids of numbers. Which pyramid represents the following food chain? So grass. In terms of numbers, there's going to be a lot of grass, isn't there? We will have loads and loads of grass, so that's going to be nice and wide at the bottom. The number of rabbits feeding on that grass is going to be a little bit less. The number of foxes feeding on those rabbits is going to be a little bit less again. And the number of fleas that are living on the fox. Now, because fleas are so small and there's going to be lots and lots of them living on each fox, that means the number of fleas is actually greater than the number of foxes. So we're going to have that kind of odd shaped pyramid where we've got the top tier being wider than the tier below it. And the one which fits that description of grass being wide, rabbit narrower, fox narrower, and then fleas wider again is pyramid A. Okay. That one for B there, that's the classic one for if it's organisms that live on a producer that's a singular producer, like a tree or something similar. C is more of your classic pyramid shape, which you tend to see in a lot of food chains, and which we would see for our pyramids of energy. And D would be a very unusual food chain. Not impossible, but very unusual to see that one. Okay, 23. Researchers investigated natural selection in a population of owls. The graph shows the changes to the percentage of owls with brown feathers over time. Okay, so we see with various years there, the percentage of owls that have brown feathers. So which of the following statements is correct? The percentage of owls with brown feathers. Now for this type of question, I would always suggest just going through each of the options and eliminating them as you go until you find one which is correct based on the data in the graph. So does the percentage of owls double between 1993 and 2003? Let's have a check. So 1993, it's up at 21, and 2003, it's up at 42. Is 42 double 21? It is indeed. Okay, so we're pretty sure that's the answer, but I always tell my students to go through all the other answers and make sure that they are wrong, and you know that for sure. So does it increase continually between 83 and 2008? Well, no, we can see between 83 and 88, it actually goes down, so we can eliminate B. Does it increase more between 88 and 93 than any other 5-year period? If we look at 88 to 93, we can see, so just from the naked eye, we can see the gradient here is not as steep as the gradient between 1998 and 2003. I would always double check that by actually counting the difference and making sure that 98 to 2003 is a greater increase, but it is in this case. Yeah, as I say, looking at the gradients is a quick way of just making sure of that. And D increases less between 1993 and 1998 than it does between 2003 and 2008. So between 93 and 98, it goes up from 21 to 27, so that's an increase of 6. And between 2003 and 2008, it goes up by 42 to 46, so that's an increase of 4. So actually it increases more between 93 and 98 than it does between 2003 and 2008. So that one is definitely wrong as well. So the answer is definitely A. Although it seems like a lot more work, just ruling out those last 3 is a sensible approach in your exam. Question 24. Nematode worms are used by farmers to prey upon insects that damage their crops. This method used to increase crop yield is an example of. So a nematode worm is a natural predator of these insects, okay, because it says to prey upon them. So it's a natural predator. And the term that we use to describe the natural predator of an organism that's introduced as an alternative to pesticides is a biological control. Okay, so the answer is going to be B there. 5. The number of light and dark peppered moths in a woodland were counted over a 5-year period. The results are shown in the table. So we can see the average number of light moths. We can see they're going down by a few, by about 20 or 40 every year. The average number of dark variety moths is increasing for every year, again going up by 40, 60, 20 over the course of those 5 years. Anyway, the percentage of moths counted in year 4 that were the dark variety was. So let's have a look at year 4. In year 4, we have a total of 180 plus 120. That's 300 moths. Of those, 80 of those 300 are dark. So we have to work out 180 out of 300 as a percentage. So if you do 180 divided by 300 times by 100, you should get 60%. So the answer for this final question would be D. Hope that was useful to run through the paper there, and stay tuned for part 2, which will be coming up soon.